Jake Feraldez, Kyle Reichman.
In our experiment, we are taking wild-type C. elegans and feeding them a bacteria that makes double-stranded RNA, or dsRNA.
The dsRNA is going to trigger the RNAi to shut off a gene. Our gene is gene letter D.
To get the worms from the starter plate onto the plate with the bacteria, we chunked the worms.
These worms that you see on screen are the wild type C. elegans.
They tend to move around in an S shape, both forwards and backwards, all the while twitching their head looking for food in the bacteria.
As you have been watching lots of worms, here's a picture of a dog to brighten your spirits. These are the eggs. L1s, L2s, L3s, L4s, and adults. You can tell they are adults because they have eggs inside of them.
This is our plate 4 days after the chunking.
We did not have good mutation results.
These worms were from a different plate.
These worms were affected and looked short and very fat.
The mutated worms were not acting any different than the wild types and were only affected in their appearance.
So our gene was called DPY10. The gene produces a cuticle collagen protein.
This protein gives the worm's skin elasticity so that it can elongate as it grows.
The mutated worms who end up with this gene silence turn out to look short and fat. Without the protein the GPY10 gene makes, the worms cannot grow any longer.
When we fed the worms the dsRNA, it tricked the RNAi system, which made the RNAi destroy the RNA that codes for the protein needed for these worms to be able to grow properly.
This experiment was a fun and interesting way to learn about the RNAi system.
After doing this experiment, we have not only gained more knowledge on the RNAi pathway, but also had an opportunity to genetically modify some organisms.
This is an intro into a new and progressing field that can only provide opportunity in the future.